Good morning, everybody, and welcome to another day of 𝗚liche and another day of Vitrolife Academy Studio. We're kicking off today's session with Robbie Kerr, who will be talking about labware in the IVF development. Thank you, Robbie.
Okay, good morning everyone, and it's great to be here at 𝗚liche in Milan and to talk to you this morning about my experiences with Vitrolife's labware and the importance of using good quality labware in your IVF program. So, we know that during the IVF process, our gametes and embryos are going to come into contact with many different materials, from needles at oocyte collection, oils, culture media, and especially dishes and plasticware. So, if we think of the IVF process as a chain, we're only ever as strong as the weakest link in that chain, and it only takes one suboptimal material to bring that entire chain down. So, if we think specifically around our challenges with plastic disposables, we have to find the right quality, as on average, from egg collection through to transfer, our gametes and embryos are going to come into contact with more than 15 different items. And these variables can all have an impact on the outcome, and it's critical that we avoid toxic and suboptimal components here, and that we consider the temperature profiles and the design of those plastics. So, I like to break down the importance of plastics into three components. So we have to have low toxicity, we have to have a nice unique design and temperature profile, and we have to have the appropriate certification on those plastics. Now, each of these components has equal importance in getting that right in your system. So, in David Gardner's former lab at the Colorado Center for Reproductive Medicine, they tested all of the supplies that entered the lab using a mouse embryo assay, and they detected that 1 in 4 of the contact materials entering the laboratory were potentially reprotoxic, and this equates to roughly 25% of all plastics and consumables used. And then a further study carried out by 𝗚cie 𝗂𝗿 in 2019, where they looked at 36 different product types from 72 different brands. They tested these products in terms of patient-related products that were used during the egg collection procedure, for example, gloves, etc. And then they had those products which were classified as direct contact with gametes and embryos. And these would cover things such as plasticware, pipettes, and dishes. And using the human sperm survival assay, they found that in those products used for patient-related procedures, almost 42% of these showed reprotoxicity. When they looked at those products used which were direct contact with gametes and embryos, they found that 10% of these products were reprotoxic. Interestingly, they also found that there were lot-to-lot differences were detected for these products, and critically that this toxicity could be transferred from one product to another. So let's put that into context. So if 25% of the consumables we use in our IVF labs have the potential of introducing reprotoxicity, what are the chances that our culture system will be unaffected by this? So where we have one contact material, we have a 75% chance of having an unaffected culture system. As the number of these potentially toxic materials increases, we can see that the chances of your culture system being unaffected starts to decrease quite dramatically. And as we get up to large numbers such as 10 contact materials, we only have a 6% chance of our culture system being unaffected. Now, I think it's important to think about this, that when we talk about an effect on the culture system, this may not necessarily mean that the gametes and embryos degenerate, but rather that we see a subtle cumulative effect over time as we have an accumulation of toxicity in the system, which could affect fertilization rates and cause disturbances in embryo development. So, if we think about traditional IVF plasticware, many of these dishes were designed rather for tissue culture applications. They were never designed specifically for IVF use. And this means they have a design which is not the ideal that we would require. So many traditional plastics would use in an IVF lab would have an air gap between the base of the culture dish and the heated surface. This means that laboratories need to adjust the set points on their heated stages to accommodate for this air gap to achieve a consistent temperature in the dish. Now, where you start to use mix and match from many different suppliers, these plastics will all have a different profile and different thermodynamics, which means if you've calibrated your stage for one dish, it's not necessarily going to be the same temperature in another manufacturer's dish type. So we looked at switching to the Vitrolife plastics in our last lab to solve this problem, because each of the Vitrolife dishes has been specifically designed and is intended to be used for IVF processes. We have a unique space for patient labelling, which is away from the culture area, so you have no interference with your imaging on the microscope. They have a unique feature that they have the tapered edges towards the periphery of the wells, which means it's easier to identify your eggs and embryos within the dish. But most importantly, they have a homogeneous temperature profile across the entire range. So looking at the process in my own previous laboratory, we mapped our heated stage using a PT100 probe integrated into one of the Vitrolife dishes. And we took measurements on our heated stage to the left and right hand side of the microscope with the lid on, and then in the centre under the microscope with the lid off, as would be the case when you're carrying out any procedures. And we could see that as my stages had initially been calibrated for a conventional 60 centimetre round dish, which would be a tissue culture based design, we could see that actually in the critical area under the microscope, mapping with a Vitrolife dish, we actually had a temperature of 37.8 in the central area. So it was key that we needed to make an adjustment here. So, and when we look at this in terms of the temperature stability in the Vitrolife plasticware compared to some traditional plastics, we can see in these graphs that the Vitrolife plasticware holds a much more stable temperature in a dish with the lid removed with no oil on the heated stage. So in the next slide here, I have some thermodynamic profiles from the dishes. So if we look at the Vitrolife square dishes, we can see that once the stage is calibrated to 37 degrees C, we're able to see that we have a consistent temperature profile compared to some traditional IVF plastics, which you can see the temperature is clearly sitting below 37 in these cases. So the great thing about the plastics is it's a one-step process. You calibrate the stage once to 37 and then make your measurements, and then you are then able to achieve this consistent profile across the entire range, regardless of the dish type you use for that process. And then the final aspect I spoke about earlier was that of certification of the labware. Now this is critical because there are two types of certification out there. You will have plasticware that is certified for IVF use, and you will have plastics that are IVD certified. Now, these are very, very different classifications. So, any plastic that's certified for IVF is intended for the culture of gametes and embryos outside of the body and is safe for the reintroduction of those gametes and embryos. An IVD certified plastic, on the other hand, will be certified for the culture of gametes and embryos outside the body, but only for examination purposes. They should not be reintroduced into the body. And this is critical because both of these classifications have different regulations that they must adhere to in their manufacture. Any plastic that's certified for IVF use will have to adhere to the MDD, MDR regulations, whereas an IVD diagnostic device will be the IVD regulations, which are very different, particularly in terms of the manufacture of these plastics. So if we look at this in a little bit more detail, the manufacturing processes for these devices are very, very different. So where you have a device that has been certified for IVF, this will have used non-embryo toxic materials in its manufacture. It will have a strict control of temperature to reduce any heat-induced toxicity in its manufacture. There'll be a single batch bioassay used for all of the raw materials that are used in the manufacture of that plastic, and it will be certified by the appropriate regulatory authorities. And this results in an embryo-friendly product. Low-grade plastics, which are not intended for IVF use, would use plasticizers and mold release agents routinely in their manufacture. And these are often petroleum waxes, which we know can be embryotoxic. They'll use high temperature manufacture, which increases the speed and capacity of manufacture. They'll mix different raw materials of different quality, and they'll be sterilized at high dose. And this equates to a low cost production of high volume product, which is potentially embryotoxic. Now, in the UK, obviously the Human Fertilisation and Embryology Authority, they recommend that all IVF labs must use IVF certified CE marked products wherever possible. The only exception to this is if there is no CE marked product available. And this is also encompassed into the 𝗚cie good practice regulations as well. So, if we look at some data in terms of evaluation, so in my previous laboratory when we introduced our Vitrolife plastics to the system, we did a retrospective analysis of our data pre-implementation and compared the clinical pregnancy rates to the 6 months after the implementation. We saw a consistent 2 p.m. fertilization rate, good 𝗚cie survival rates with using the plastics, and we saw an increase in clinical pregnancy rate. Now, when we compare this to the external clinical validation carried out by Vitrolife, once again, this was real-life clinical data collected from 3 different centres where they compared conventional plastics against the Vitrolife plastics. And they evaluated treatment outcome to a matched historical control. And once again, they were able to see a very similar increase in clinical pregnancy rate as I observed in my own laboratory at that time. So I think this validates, the importance of using good quality plastics in your system. So to summarise, I think the labware covers the entire process from oocyte collection all the way through to transfer. Each dish has been specifically designed for its intended use. It is a uniform, homogeneous temperature profile across the entire range, and this really helps with improving the process control within your laboratory. We have exceptionally high quality control on the products that each batch is extensively tested in terms of the materials used in its manufacture, the in-process testing, and the final products that are released to you, the customer. And each batch of plastic meets the strict regulatory requirements for both CE marking for IVF and the FDA regulations for IVF. Thank you.